Okay, tomorrow is another important lab in terms of identification of your unknown. As a matter of fact, really after tomorrow, you'll have virtually all the information you need to start doing the identification. In lab number 16, we will be doing a couple of things that could kind of supplement your information, but the basic stuff you need for your keying will be done on Wednesday, inoculated, and then Friday, we will do like we did last Friday and record results and look at your results. So it's important to have a good working culture with a fair amount of growth on it because there's a fair amount of inoculation to do. You'll work with partners as you did last time doing the known organisms, pretty much same thing we did last time. Different tests. Any questions? So, what are the four compartments of the immune system? RES, extracellular fluid, lymphatic system, and the blood. Okay? And all of those are interconnected in one way or another. And so if any infectious agent gets into any of those particular compartments, the response is translated to and through the other compartments as well. We talked about the functions of the immune system. For our purposes, the major two ones that are relevant are, the lymphatic system. The lymphatic system helps to resolve or correct edema, swelling. Swelling is one of the results of inflammation, and inflammation is one of your important second lines of defense that we'll talk about probably next Monday. That's about when we'll probably get to inflammation. So you need a system that helps to resolve that inflammatory response, and the lymphatic system is crucial there. But ultimately, most of the interactions that take place between pathogens and immune cells take place within the lymphatic system, and that's where most of those interactions take place. I think most people incorrectly assume when they're learning about the immune system that most of the important reactions take place in the blood. Actually, no. Most of the important interactions take place within the lymphatic system. So we start differentiating types of lymphatic tissue into primary and secondary types of tissues. The primary tissues are the bone marrow and the thymus. And the bone marrow is responsible for producing the educated B lymphocytes, and the thymus is responsible for producing the educated T lymphocytes. What do we mean by education? What do you have if you're an educated lymphocyte that you didn't have before you were educated? You have a particular receptor for a particular antigen. So that process of development, which happens in the bone marrow and the thymus, involves these receptors being built and then assembled on the surface of those particular cells. So genes have to be transcribed, proteins have to be translated, proteins have to be transported out of the cytoplasm onto the cell surface, and then you've got these nice receptors, each of which is specific for a particular antigen. Okay? Once you have become educated, doesn't mean you are active. Those educated immune cells are sometimes referred to as being naive. They know what they're doing, but they're not doing anything yet. So what does it really take to turn these naive B lymphocytes into cells that do something? What kind of stimulus do they need? What turns that educated B lymphocyte with its feet propped up into an actively dividing and participating cell in the immune system? Getting bound by your particular antigen you are paired up with. And typically that is the result of an antigen-presenting cell showing you that antigen. Typically it is. There are instances where the antigen can directly stimulate the B cell or the T cell. Most likely it involves antigen presentation. Cells that do something are called effector cells. The major B effector cell is a plasma cell. And what does that plasma cell do? What does that B lymphocyte do in your immune response? Remember that B lymphocytes are involved in humoral immunity? What's happening in humoral immunity? What are the B lymphocytes doing to help protect you? They're secreting something, aren't they? What are they secreting? They're secreting the antibody. So those are the antibody-producing cells. Once a cell has become stimulated by antigen and starts differentiating into plasma secreting antibody cells, or antibody secreting plasma cells, sorry, another type of cell that's produced during this immune response is a memory cell. So this is where you get your immunological memory. You've seen the threat before. And next time that these memory cells are stimulated by antigen, they can respond much more rapidly than initially the naive cells could. So that's why immunization is so important. You want to prime your immune system to create immunological memory, and then if you're exposed to that real pathogen, then these cells respond really fast to eliminate it. If you've never seen it before, if you've never been immunized, your first response will be by these naive cells. And it takes time for them to gear up to start making plasma cells and secreting antibody. And by that time, the pathogen might have gained the upper hand. And that's bad. Once these naive cells are produced, they will then migrate into one or more of these secondary lymphoid tissues. And that's really where they sit and wait to encounter their particular antigen. So B cells are born in the bone marrow. They're educated in the bone marrow as well, make their receptors, and then they migrate out to these secondary lymphoid tissues and sit there and wait to do something. Also remember that an important thing that happens in the bone marrow for B lymphocytes is that self-reacting cells are eliminated. So you don't get that autoimmune type of reaction. One of the things we're going to come back and talk about was the question that Arthur raised, and that was issues regarding making these particular receptors. And how do you make, billions of different type of receptors in these particular cells? Some very interesting genetics happen within the bone marrow during this education process. And one of the important things that happens is a very unique and interesting phenomenon called gene shuffling. And the only place you find gene shuffling occurring is within these cells that are going to give rise to the B and the T lymphocytes. Okay, the other primary lymphoid tissue is the thymus. T lymphocytes are also born in the bone marrow. That's where the process of forming the initial immature T lymphocyte takes place, is in the bone marrow. Then those precursors of T lymphocytes migrate to the thymus, and that's where they become educated. So T lymphocytes build their receptors in the thymus, where B lymphocytes build them in the bone marrow where they're born. So you can think of B lymphocytes as kind of being homeschooled. T lymphocytes go off to school. They go to a new location. Once they have developed their particular receptor, they migrate out into the secondary lymphoid tissue as well. And then they sit there and wait. Wait for what? Wait to be presented their particular antigen. And if they're presented their particular antigen, they're activated. And they start turning into effector cells and memory cells as well. But remember, this branch of immunity is the cell-mediated immunity. So we're not talking about secreting antibody. That's humoral immunity. Remember, in cell-mediated immunity, we're destroying abnormal host cells, like a cancer cell or a virus-infected cell. So what are our effector cells here? Well, one of the major players is a type of cell called a T killer cell. And the T killer cell literally punches holes in the surface of the abnormal host cell and kills it. Another type of T effector cell that can be produced is a suppressor cell. And as you might expect, what does this do? This helps to shut down the immune response when the threat is eliminated. It's kind of nice to have an off switch as well as an on switch in a particular cell. And then the third effector cell is the T helper. What does the T helper do? That helps the B lymphocyte to secrete antibody. So once your immune system has started to develop when you're young, eventually you have populations of educated but naive B and T cells sitting out there in all of these different regions of the body that make up your lymphatic system. In the nodes, in the spleen, in the appendix, in the malt, and in the gall. And then they just sit there and wait to be stimulated. Into these, causing humoral immunity, or into these, causing cell-mediated immunity. Now, the thymus is really large in the developing fetus and once a baby is born. Over time, as that child gets a little bit older, the thymus starts to shrink and wither and eventually becomes non-functional in adulthood. But by that time, the immature T cells that were produced in the bone marrow have migrated to the thymus and become educated, and then these secondary lymphatic tissues have been seeded with those naive T cells. So at some point in time, the thymus kind of loses its relevancy. It really, you don't need it anymore. It because we've seeded these particular locations, and now they're good to go. Anybody ever dissected a fetal pig? One of the things, if you dissect a fetal pig and you do your thoracic incision, is that when you make that thoracic incision and you peel away the skin and the muscle, you see all this granular looking tissue lying on top of the trachea and sometimes flopping over on top of the heart. That's the thymus, okay? And it's huge in that fetus. It's really very important in establishing your immune system, especially your T lymphocytes, to begin with. But eventually it shrinks and shrivels up because its job is done by early childhood. Okay, what are some of these other lymphoid tissues? MALT stands for mucosal associated lymphatic tissue. You find concentrations of MALT in the thoracic cavity and in the tonsils. So this idea that tonsils are entirely expendable and are a structure that has no function is really not entirely true. Okay, it's one of your kind of backup storehouses of immune cells. Now, can you get along without your tonsils? Yes, you can. Does it immunocompromise you a little bit? Potentially, it does. But if those tonsils become chronically infected, you're probably better off having them removed than having that particular backup system. Remember that I told you that one of the characteristics of the immune system is redundancy? And there are backups to backups to backups. So if you have to remove one for a legitimate purpose, it's probably not going to be devastating. GALT stands for gut-associated lymphatic tissue. And one of the types of gut lymphatic tissue that's fairly abundant in the gut are patches of cells called the Peyer's patches. Those are associated with the ileum of the small intestine. So you get a really high concentration of these lymphatic tissues in both the thoracic cavity, in the oral cavity, and in the abdominal cavity. Well, those are three primary portals of entrance for pathogens. And so having those lymphatic tissues there with their resident populations of B cells and T cells sitting there waiting is a pretty good strategy. Spleen is another secondary lymphoid tissue. The spleen has really two functional parts to it, called the red pulp and the white pulp. The red pulp is primarily involved in the recycling and destruction of red blood cells. In some respects, the spleen is kind of a filter for the circulatory system. Why do we need to recycle red blood cells? Remember, red blood cells only have a finite lifetime. They last about 3 months. They're non-nucleated. Eventually, they break down. And recycling the hemoglobin and the iron, particularly from the hemoglobin, is an important function of that red pulp of the spleen. The white pulp has high concentrations of both B and T lymphocytes. So if infectious agents get into the blood will flow through the spleen, and there the infectious agents could then be exposed to those waiting B and T lymphocytes. You tend to find B lymphocytes in T lymphocytes in different regions of the white pulp. So Some parts of the white pulp have mainly B lymphocytes, some have mainly T lymphocytes. The real workhorse of the lymphatic system, however, are the lymph nodes. And this particular slide shows you the relationship between the lymphatic system, the circulatory system, and the extracellular tissue fluid. So here we've got some lymphatic vessels. They're the ones that absorb a lot of that fluid that might have leaked out into the lymphatic system. Remember that lymph flow is one way, and all the lymphatic fluid eventually moves back toward the heart. The lymphatic ducts empty into the right subclavian artery and eventually get into the circulatory system. It's important to have muscular contraction to push the fluid through the lymph. So there's no heart that pumps lymphatic fluid like the heart does for the circulatory system. Lymphatic fluid moving back to the heart all involves muscular contraction. Lymph nodes are these swollen regions into which several different lymphatic vessels from different parts of the body empty. Again, you can think of it as kind of a filtering station. Structurally, there are two functional parts to the lymph node. There is this inner medulla region, and there is this outer cortical region. B cells tend to be found more in this outer cortical region of the lymph node, and T lymphocytes tend to be more localized in this inner medulla region. But if there are infectious agents in this tissue fluid somewhere in the body, eventually that tissue fluid will be reabsorbed into lymphatic vessels. And as those lymphatic vessels move back toward the heart, they will pass through one or more filtering stations, the lymph node, and there those particular infectious agents will encounter B and T lymphocytes that are hopefully paired with them and are able to bind to them and react and mount an immune response against them. So that's the lymphatic portion of the circulatory system. How about blood? If we take whole blood and centrifuge it, it separates into two basic components, a liquid component called plasma and a cellular component containing the cells. The white blood cells tend to form this little buffy coat region that's at the interface between the liquid plasma and the red blood cells and platelets. So this mixture down here is mainly red blood cells, but there are platelets in there as well. So here's where we find a lot of our white blood cells in this particular buffy coat. What is plasma? Well, plasma is mainly water. Remember that blood is atypical connective tissue, and in this particular connective tissue, the cells are suspended not in a solid matrix, like it is in cartilage, for example, but instead a liquid matrix, which is water. There are a variety of proteins that we find in the plasma of blood. We've mentioned, for example, transferrin. That is one of those iron-containing proteins. There are a variety of clotting proteins circulating around in your blood. In response to injury, those clotting proteins can be activated and form a cascade reaction where one activated protein activates another and activates another and activates another. And ultimately, as a result of the cascade, an insoluble mesh-like protein is activated called thrombin. And that insoluble thrombin protein starts trapping red blood cells in kind of a meshwork and forms a clot. And clotting is part of your defense mechanism. It can trap pathogens. It can help to temporarily plug an injured vessel until healing of that vessel has time to take place. So if we take blood and we just let it sit, when it's exposed to oxygen, that blood will tend to clot. The clot will trap the formed cellular elements, the red blood cells, the white blood cells, the platelets, and then a liquid will be left over which contains all of the proteins that are in plasma except the clotting proteins. So serum is plasma minus clotting proteins. Another important set of proteins that are found in the blood plasma are complement proteins. In response to infection, complement proteins interact with one another, also in a cascading fashion. So one complement protein becomes activated, it activates another one, that activates another one. And the net result of the activation of the complement system is the pathogenic cells are lysed. They get holes punched in them. Another really important type of protein that's found in the blood serum are gamma globulins. Antibodies belong to this class of protein that are globular in nature, and particularly the gamma class are the antibodies. So gamma globulins are antibodies. So there's some really important proteins that are involved in your body's defense systems that are constantly circulating around in the liquid portion of the blood. But obviously, too, the cellular elements are incredibly important in a functioning immune system. Where do these cellular elements come from? Well, there are stem cells that are present in the bone marrow. And your bone marrow, through your entire life, is constantly cranking out erythrocytes, leukocytes, red blood cells, white blood cells, and the megakaryocytes, which are the ones that fragment later and produce the platelets. So stem cells in your bone marrow are constantly resupplying red blood cells, white blood cells, and platelets, and they're then entering the blood. This process of producing blood cells is called hemopoiesis. The primary site of hemopoiesis in an adult is the bone marrow, particularly of the long bones of the body. So over here on the right, you can see this bone marrow is where all the stem cells are that are constantly cranking out precursors to red blood cells, white blood cells, and platelets. So there's a lot of hemopoietic activity taking place in these regions of the body. That's not the way it starts out. In a fetus, before the bones begin to develop, hemopoiesis begins in the embryonic yolk sac. And so that's where initially our stem cells are that produce the very first blood cells. In a period between a 5-week embryo and an 8-week embryo, that yolk sac is eventually absorbed and becomes non-functional. The liver then temporarily takes over hemopoiesis in a very young embryo and continues to produce stem cells that give rise to blood cells until the skeletal structure starts to develop. And once the skeletal structure starts to develop and the long bones start to form, then hemopoiesis is taking place within those particular locations. We're only going to be interested in the white blood cell elements. There are different paths of differentiation in the bone marrow for red blood cells, white blood cells, and platelets. But we're only going to be interested in the white blood cell portion of hemopoiesis. The differentiation pathway of white blood cell hemopoiesis results in several different types of white blood cells. I mentioned a couple of them already. B lymphocytes are one type of white blood cell. T lymphocytes are another type. Macrophages are another type. Dendritic cells are another type. And that's not all the players. Okay, we're going to introduce you to a few more players, a few more products of hemopoiesis. All the white blood cells can be grouped into two major categories: granulocytes and agranulocytes. Granulocytes have very prominent cytoplasmic granules. And one of those cytoplasmic granules is a lysosome. There are other granules as well, but lysosomes are one of the major cytoplasmic granules in these granulocytic cells. Agranulocytes are not really lacking these granules, they're just very small. They don't appear to be present if you look at them microscopically. Another difference in these particular types of cells is that the granulocytes tend to have more lobed nuclei. Agranulocytes tend to have a little bit more circular nuclei. So there's some morphological differences in the nuclei of these two particular types of cells. Another way to differentiate granulocytes is by doing a differential stain called a Wright stain. I don't know if you remember, but one of the things about differential stains is that they involve the use of multiple staining reagents in the staining procedure. So the Gram stain is a differential stain. The Gram stain uses crystal violet, it uses saffron. Okay? And it breaks things up into two particular categories, the staining results, gram positive, gram negative. Endospore stain is a structural stain. It typically involves one staining reagent, malachite green. The two components of Wright stain are eosin and methylene blue. And eosin is kind of a reddish stain. Methylene blue is obviously a bluish stain. Methylene blue is a cationic or positively charged stain, a basic stain. Eosin is an acidic stain, a negatively charged. Remember, most of our stains in the lab are positively charged, basic stains, crystal violet, methylene blue. Acidic stains we used were nigrosin, and in the capsule stain, we used Congo red, which was an acidic stain. So different charges. These particular stains tend to bind to the substances in the granules of these cells. Well, as it turns out, within this group of granulocytic cells, there are different types of granulocytic cells which have different granules with different compositions. And so these staining reagents are going to react with some of these cells differently than they do others. The three types of granulocytic cells that we can differentiate between using the Wright stain are the neutrophils, the basophils, and the eosinophils. Now, there are a couple of numbers that I do want you to keep in mind for contextual purposes. One of the numbers that I want you to keep in mind is a rough approximation. 2/3, 1/3. If you take all of the white blood cells in your body under normal conditions, at any given time, about 2/3 of them will be granulocytes, and about 1/3 of them will be agranulocytes. Within this 2/3 population, what's the breakdown between relative numbers of neutrophils, basophils, and eosinophils? By far, there are more neutrophils than any other type of granulocytic cell. The number of basophils is very small. The number of eosinophils is also small, but it's greater than the number of basophils. So rank would be mostly neutrophils, a few eosinophils, and very, very few basophils. So what does a neutrophil look like following a right stain? Remember, these are granulocytic cells, so they're going to have visible cytoplasmic granules. And those granules are going to take up components of the right stain. The cytoplasmic granules of the neutrophils take up both components of the right stain. So they take up the reddish and the bluish stains. The nature of their granules binds both acidic and basic dyes. So what color are their granules? They tend to be more of a lavender, light purple color. The nuclei tend to be highly lobed in nature. Oftentimes, there are kind of two distinct lobes that you can see. I'm sorry, more than two. There are multiple lobes that you can see in a lot of those nuclei. If you see this bottom diagram down here, okay, this multi-lobe nucleus here. So in a right stain, these would be the red blood cells, and then this would be one of the leukocytes. This would be one of the white blood cells. Which one? This would be the neutrophil. How do you know? Because of the lavender granules and the highly lobed nucleus. Those tend to be the first line of defense against infectious agents. And what are they doing for you? Phagocytosis. These particular cells are adapted to be monster phagocytes. Chew things up and break them down. They're mobile. And they can crawl around in the body. They actually spend very little time in the blood. Now, they are originally produced in the bone marrow and released into the circulatory system. But they only spend a few hours in the blood, and then they migrate out into the surrounding tissues, like the RES system, and kind of patrol around and look for things that don't belong. Neutrophils have some receptors on their surface called toll-like receptors. And these receptors match PAMPs. PAMPs stands for, I already told you this, but pathogen-associated molecular patterns. So peptidoglycan is a PAMP. And these neutrophils have some receptors on their surface that recognize this general category of molecule, peptidoglycan. And if they encounter something that has peptidoglycan on its surface, then they tend to respond to that and phagocytize it. So there's some level of group specificity, but not individual specificity. They're not looking for a particular unique antigen. They're looking for this kind of category of antigen that represents pathogen. There are also certain receptors on the surface of a neutrophil for antibodies. And if those antibodies are coating a pathogen, then that means that the neutrophil can recognize the pathogen because it's coated by this antibody. And therefore it's going to gobble it up. The antibody is serving as kind of the kick me sign I talked about. It's attaching to the pathogen and going, hey, this doesn't belong here. And then the neutrophil can recognize that antibody and bind to it, and therefore it's bound the pathogen. Those antibodies that help to stimulate phagocytosis are called opsonins. So if your immune response produces antibodies that are opsonins, they coat the pathogen, and then the neutrophil looks for that particular antibody, binds to it, and therefore it's bound the pathogen, and then it can gobble it up. Neutrophils are different from macrophages in two important ways. They don't process and display antigens like macrophages do. So neutrophils are not antigen-presenting cells. Neutrophils also don't produce lymphokines. Macrophages do. What are lymphokines? They're chemical signals that get other immune cells involved in the process. So neutrophils are kind of the foot soldiers of the Army of Defense. They're the ones that are usually arriving at the infection first. They're attracted by substances that the pathogen produces. So chemotaxis occurs, the pathogen actually attracts the neutrophil unwittingly. Substances that are produced during inflammation also attract neutrophils. What do they start doing? They start phagocytizing the pathogen and trying to destroy it. What does the pathogen do? It might produce substances like leukocidins, which kill the neutrophils. And so the area of infection can literally become this gross battlefield where there's a whole bunch of dead bacteria, a whole bunch of dead neutrophils, and that accumulation of the killed white blood cells forms those pussy pockets that are associated with infection. But the neutrophils have limitations in terms of what they can do. They're pretty much these grunts of the immune system that can do one thing, engulf and phagocytize, but they don't have the sophistication to communicate with other cells. They don't present antigens to other cells. You can kind of think of the macrophages as being more of the officers of the army. And they've got a lot more tools that they can use in the immune response. Next time we'll talk a little bit about basophils and eosinophils. They have a moderate role that they play in immunity as far as we're concerned.
Probably